to take a look at the internal structure of the sun, for example. Suppose let me take a 3D diagram. Hold on. Okay, anyways, see. So say that this is the core of the sun. Okay, core of the sun. Now the core of the sun is also divided into different parts. Basically, this is going to be the centermost region of the sun. Right now, let me write the topic also. We are learning about the structure of sun. Structure of sun. Basically, it's different layers. So you have the innermost layer, the innermost region of the sun, that is the core. Now, inside the core, the nuclear fusion reaction occurs. What I'm saying that inside the core of the sun, the nuclear fusion reaction occurs. So you have the core, innermost layer. Okay. Next, this is the place where nuclear fusion is occurring. Getting it? That is one thing. So basically, in this, what happens in this nuclear fusion reaction, the nucleus of the hydrogen would be combining together to form helium, and thereby in this process, lots of energy will be released. Getting what I'm trying to say, everyone?
Yes, sir.
So in the fusion reaction that is occurring in the core of the sun, basically nucleus of the hydrogen would be combining. So hydrogen plus hydrogen would be reacting to form helium. And in this process, tremendous amount of energy is being released. So energy is released. So that is all about the innermost part. Okay. Furthermore, this innermost layer of the sun, what do you guys say? Is it solid in nature? Is it going to be gaseous or liquid? Any idea?
Solid.
No, it is gaseous in nature. Okay, now Abdullah, by the way, right now we are taking up a doubt. Chakrika had doubt in the structure of the sun, so that is what we are doing. So this is made up of gas. So another point about the core, it is made up of gas. Obviously, one more important thing, that this is going to be the hottest part of the sun, isn't it? This is hottest part of sun. Okay, hottest part of the sun. And I think the temperature here can reach somewhere between 14 to 15 million degrees Celsius. Okay, now? So just imagine how intense temperature this is. Okay, now just like 50 degrees Celsius feels so warm. So if it is 15 million, about 15 million, I'm not exactly sure, but about around 15 million degrees Celsius. Getting it? So temperature can reach as high as 15 million degree Celsius. So that is about all of this.
Sir,
Yes.
good evening, sir. Sir,
Yeah.
actually, your voice is cracking.
Voice is cracking.
Yes.
Is that the case with everyone? Am I audible to others properly?
Yes, sir, you are audible.
Okay, sir.
Then it might be some network issue with you. 1 minute, let me.
Sir, not every time, but like in between, sometimes it's cracking.
Okay. See. Next, in the core, we see that further, like right after the core, you have some other layers also. Okay, now. So after the core, you have the zone where radiation is being released. Okay, now. So this is the core, and right after the core, the next layer this is being called as the radiation zone, basically. Okay, so basically the energy that is being created in the core will be released through this radiation zone. Getting what I'm trying to say, Chakrika and everyone? That is the radiation zone. So after core, we have the radiation zone. So in radiation zone, basically, energy from the core is being kind of transported through this. It goes through this layer. Okay, now? So this is the second layer. Okay, we are counting it from the core. So thereby it is the second layer. Okay. Furthermore, energy is transported, or energy from core is transported through this. So far it is clear, Chakrika and everyone?
Yes, sir.
Okay. Now after this, we have another zone that is called as chromosphere. That is called as chromosphere. Getting it? So this layer, suppose this one is the chromosphere. Okay. Not talking about the chromosphere, just 1 minute. Even before the chromosphere, now we have one more layer. 1 minute. Even before this, you have the convection zone. Hold on. So after this, you have this layer that we are calling as convection zone. So that's first, that's the second zone, and the third zone here is the convection zone. Okay. As for the convection zone, this, in terms of the interior layers of the sun, this will be the outermost layer out of all the interior layers. Getting it? Furthermore, sun also has its magnetic field, just like the earth has its magnetic field. So similarly, we see that sun also has its magnetic field. Getting it, everyone? So the magnetic field of the sun is arising out of this convection zone. Getting it, everyone? Chakrika and everyone?
Yes,
So
sir.
sun's magnetic field arises from this zone. Okay. After the convection zone, the fourth zone now is basically for another layer, basically is the chromosphere.
Also,
Okay.
isn't it photosphere?
Photosphere.
Yes.
Photosphere. Photosphere, it's the visible layer of the visible surface of the sun, I think, basically.
So, sir, is it after chromosphere or like before chromosphere?
No, you are actually right. You are actually right. It is before the chromosphere, actually. Good. Yeah. Before chromosphere, you have this protosphere. Very good. So you have got protosphere, which is basically the visible surface of sun. After the protosphere, you have got this chromosphere layer. Okay, you have got the chromosphere. Okay. Now here the temperature can increase. Basically, it is not fixed. On different parts of the sun, the temperature in this layer would be varying basically. Okay. After this, basically, after the chromosphere, you'd be having the outermost layer of the sun basically, or basically it's not the surface, but it's the atmosphere of the sun. Getting it? So that, I think I have mentioned this in the previous classes also, sometime might be. So that is the corona basically. Okay, now that is the corona. So what is corona? Basically is it's the sun's outer atmosphere. Sun's outer atmosphere. So these are the different layers. Getting it, everyone? You have the core. Core further has got radiation zone in it. It has convection zone in it. Getting it? So this is the first layer. That is the core. Okay. Inside the core, I would say that radiation zone is part of the core. Convection zone is also part of the core. The second layer is the protosphere. Third is the chromosphere. And the last one, that is the outer atmosphere of the sun, that is the corona. Okay.
Yes, sir.
Shigarika, if you have written it, it would be good, or you can take a screenshot of this.
Yes, sir. I've written it down. I wrote it down.
Good. So that was your doubt. Hope it is cleared. Yeah.
Yes, sir. Thank you, sir.
Yes, you're welcome. Hmm. So as we were doing this topic in the previous class about division of microorganisms, okay. So this group of microorganisms is the protozoa. Basically, they are unicellular in nature. Some of them would be living independently, okay, like they would be able to live independently, okay, while others would be living as parasites, will be having a parasitic type of nutrition. Now, many of them would be responsible for causing diseases in humans, in animals, and even in the plants. Like, for example, we have disease like amoebic dysentery, okay, that is caused by a protozoa. Getting it? So amoebiasis, or basically amoebic dysentery, is a disease that is caused by a protozoa. Also, we see that protozoan disease, basically some of the protozoas are found to be living as parasites within the gut, also within the human body also, and they will be known for causing various kind of diseases. Particularly, we have this disease called as amoebiasis, which is also called as amoebic dysentery, and this is spread via the contaminated human fecal matter. So what will happen that when the house flies, or the domestica, basically the house flies, when it will be sitting on the infected fecal matter of the human, and when it will be sitting onto the food, so thereby it will be acting as the carrier of this parasite. Getting it? So thereby food should be kept closed. It should not be left open, so that if the flies were to sit on it, it can transmit the pathogens on it and can make the person sick. So one example is this amoebiasis, also called as amoebic dysentery, that is caused by a protozoa. Okay, that is caused by a protozoa, and the name is basically and the name of the protozoa is Anta-amoeba histolytica. Not that much important, but just know the name of the disease. Okay, and how it spreads, keep that also in mind. It is spread via the house flies. So when the house flies would be sitting on the human fecal matter, so thereby it will be the pathogens, basically the protozoa will transfer to its body, and when the housefly will sit on the food, thereby it will transmit it. That is the case. So protozoa, they are unicellular in nature. Furthermore, we see that many of the protozoa are having leg-like structures. Not exactly leg-like structures, but it will basically help them in locomotion. Like for example, amoeba, we know that amoeba have false legs in them. What do we call the false leg of amoeba? That's the pseudopodia, isn't it? So this has got pseudopodia in it. Then further you see that this paramecium also has some hair-like structure, with the help of which it will be able to do locomotion and will be able to take in food with the help of the cilia. So since they are capable of locomotion, so they are referred to as animal-like organisms. Getting it, everyone? They are referred to as animal-like organisms. Okay. Can you guys name the protozoa that causes malaria? Yeah. Name the name the protozoa that causes malaria. Anyone? Yes,
Plasmodium.
it's the plasmodium. Very good. So plasmodium causes malaria basically. So what happens? This plasmodium protozoa, it basically will be found in the infected blood of the person suffering from malaria. There's a person who is already suffering from malaria. Okay, so in his blood, particularly in the red blood cells, plasmodium will be found to be living. Okay, further, when the person will be bitten by a mosquito, so mosquito via the proboscis, by its mouth, that is called as proboscis, will be sucking the blood, and when the same mosquito will be biting a healthy person, so when it will be inserting its proboscis onto the skin of the healthy person, so in doing so, the blood will be getting mixed. Yeah. and thereby the plasmodium from the mouth of the mosquito will be transferred to the healthy body. Thereby, it will be spreading this disease. Okay. Now, important notable thing here is that is it the female mosquito or the male mosquito that sucks the blood and that spreads it?
Female mosquito.
Female mosquito.
Why is it that the female mosquitoes would be sucking blood and not the male?
Male mosquito doesn't bite. I have heard it.
Male mosquitoes does not, repeat again.
Doesn't suck the blood. Male mosquito doesn't suck the blood. I have heard.
Yes, that is correct. You have heard it correctly. Basically, female mosquitoes needs blood for reproduction to produce egg. They need rich source of right now. They need a good source of iron, protein, etc. So that they get from the blood now. So since they have to produce egg, they need high amount of nutrition. So thereby they get it by sucking the blood. In this case, particularly, there is the Anopheles species of mosquitoes. So it's the female Anopheles mosquitoes that spreads malaria. Okay. Next group of microorganisms is the smallest microorganisms of all. That was the viruses. So they are said to be so small that, say, consider you have example of this virus called as rhinoviruses that causes common cold. So if you were to have a pin, okay, small pin, so 500 million rhinoviruses can fit on the head of that small pin, right? So this is so much small in size. And here is the size comparison of different cells. And here you are given with the virus cells also, like SARS-CoV-2, HIV, influenza. So these are all three different types of viruses. In comparison to this, compare it with the lysosome. I think some of you would be aware with lysosome, yeah? Those of you who have learned the cell and its different parts, so you will find that inside the cell itself is a very small thing, right? Inside the cell, we have structures called as lysosomes. So the viruses are even smaller than part of a cell. Getting it, everyone? Here you have the E. coli bacteria, and right next to this, you have the red blood cells. So viruses are very small in size. Okay. Further, they are unicellular also, right? They are unicellular also. Okay. Now you guys answer to me, who discovered viruses for the first time? The name and the picture is given here. The person that you see here, Dmitry Ivanovsky. Okay, he is credited for discovering the first virus. Okay. Then the branch of science in which we study about viruses and the diseases that they spread, that is called as virology. Further, we have got different types of viruses based on the shape they have. So you have got polyhedral shaped virus, enveloped, helical, complex, etc. Getting it? So they are different shapes of viruses also. Now, they are very different from other type of microorganisms. Can you guys tell me how the viruses are different from other groups of microbes?
Is that inactive when they're outside a host's body?
They are like non-living organisms.
When they are outside the host cell, then they are like non-living organisms. Yeah.
They are much more.
They can
And they're much more
reproduce.
smaller to compare to all of them.
Yeah, they are very smaller in comparison of all the microorganisms. Yes, Corey?
They can reproduce themselves within a second.
Yeah, that can also be considered. Okay, so they are different from other groups.
So is it true that the viruses can be seen through an electron microscope?
Viruses can be seen through a electron microscope.
Electron microscope, yes.
Okay, yes, it can be seen. Okay, now via the electron microscope, we'll be able to see this. Okay, because the electron microscopes gives you a very high resolution image. Okay, now so thereby, like we cannot see it with the normal microscopes. We have the optical microscopes, right? So with the help of that, we cannot see it. Neither we can see it with the compound microscope. So we use basically this electron microscope. Okay. Sorry. Yeah. Getting back to this topic. So I was saying that they are microscopic, but they're different from other because they reproduce only inside the cells of the host organism. So it could be anything. It could be even a bacteria. They can grow within the cell of a bacteria also. Right? So they cannot replicate on their own. They require a host cell in order to survive and reproduce. Right? So they are considered as non-living outside of a host cell. These are some of the examples: HIV, hepatitis B, Ebola virus, adenovirus, influenza, and bacteriophage. Okay. Furthermore, there are some advantages and disadvantages, right? First, talking about the disadvantages, they will be responsible for spreading so much of infectious. They will be responsible for spreading so much of diseases, right? And getting rid of a viral disease is very difficult task, right? Now, it is observed that getting rid of a bacterial disease is, sorry, 1 minute. Yeah, I was saying that it is observed that getting rid of a bacterial disease is easier in comparison to that of the viral disease. Getting it? Because preparation of antibiotics are much easier in comparison to preparation of vaccine. Getting everyone? Okay, now. So antibiotics.
So why the virus?
Repeat again.
So why the virus is always like spread by polluted places like China, India, you know?
Okay, so you're saying that why is it that viruses spreads in countries like China or in India? See, it is
Yeah.
not very specific. Okay, no, that although we see that HMPV or the COVID viruses, right? So like some of the viruses spreads particularly in countries like in China, it is observed, but it has got to do with the climatic factor, the lifestyle, the eating habit, and all such things influences the rate at which the virus would be spreading. Getting it? No?
Yes.
Okay. So I was saying that getting rid of the viral disease is much difficult in comparison to that of a bacterial disease. Okay, because the rate at which virus would be multiplying is much faster. Okay. Also, we see that antibiotics are ineffective against viruses. Okay, no. So we see that preparation of antibiotics is also much easier in comparison to preparation of vaccine. So these are the factors. Also, the rate of transmission, the rate at which it will be spreading from infected people to healthy people is very fast in case of uh, viral diseases. Okay. Furthermore, we see that there are limited advantages of viruses also. Okay. Like for example, they will be used in the gene therapy. Okay. Or for example, if some cells in the body have to be genetically modified. So in short, I would say that basically for medical purposes, for research and medical purposes, some of the viruses can be used. So that will be its only advantage. As for talking about the vaccine, so vaccine we know basically is about the vaccine we have learned that basically they contains live or weakened versions of microorganisms or viruses in it, right? Or we can have some part of that virus or the microorganisms. We can take some protein structures out of that microorganisms, and thereby we create this vaccine, which will be administered to the person in order to generate immune in the body of that person against a particular disease. For the very first time, Dr. Edward Jenner created the first vaccine, and he basically found the first vaccine for the disease smallpox. So he basically observed that the milkmaids, okay, were infected with cowpox. The milkmaid, you understand, right? The milkmaids were infected with the cowpox. And it was later observed that now they became immune to smallpox. They were not getting infected with the smallpox. So in this manner, the discovery for the first vaccine took place. Talking about what is vaccination. So vaccination is basically the administration of a vaccine, basically giving the vaccine to help the immune system develop immunity for a disease. Okay. So vaccines, they contains microorganisms or viruses in a weakened, live or killed state. It could be protein structures or some of the parts from its body. Okay. So when it is introduced into the body of a healthy person, so the body will start to fight and kill them by producing suitable type of antibodies. Okay. And this antibody is now going to remain in your blood. So when the next time the actual virus, when the actual virus enters your body will be able to recognize it and will be able to fight it and hence protect you. Got it, everyone, so far?
Yes, sir.
Answer the given question. It's simple. Yes.
So third one, malaria.
Third one is, yeah, good. Next, which of the following disease is caused by bacteria? And name the bacteria also. Roni, you there in the class now? Roni, Aleph, everyone?
So
You guessed
tuberculosis.
it's a tuberculosis. Good. Which microorganism causes tuberculosis? Yeah, anyone remember the name? Tuberculosis is caused by a microorganism. In short, this is AMBI. Anyone who can recall the name? Yeah. Yes. AMBI, I would say. So that is basically Mycobacterium tuberculosis. Okay. So this is responsible for causing tuberculosis. Okay. What about the typhoid? Name the bacteria responsible for spreading typhoid, for causing typhoid. Yes, Alif, Abdullah. Which microorganism causes typhoid? Yes, what is it?
I think why? No, not virus. It's
Salmonella typhi. Okay,
bacteria.
no? It is Salmonella typhi. Okay, it's the bacteria that causes typhoid. Okay. We had also discussed about where the microorganism is found. So microorganisms is found almost everywhere. The air we breathe in, the water we drink, the land on which we live, right? So it is present everywhere. It is even found in the extreme conditions, like in the hot water springs, in the polar ice caps, right? In acidic water also it can be found. Further, it is present inside the body also, inside the body of humans also, in animals and plants, and even some microorganisms also have some other microorganisms living in their body. As for you have like bacteria, inside the bacteria a virus could be living. So microorganism would be found everywhere, right? Furthermore, we see that in our body particularly, we see that, okay, you guys tell me, where in the human body most of the microorganisms are found? Maximum amount of microorganisms will be found
In
in which
stomach
part? In
and
the
the digestive.
Hmm.
Large
Mhm. We can combine all of them together. Basically, the gut. Okay, now the gut has maximum number of microorganisms living in it. Okay, now it will be having a variety of bacteria would be there, protozoars would be there, etc. Okay. Now some of them are healthy also. Some of them are essential for our body. Okay, now Some of the microorganisms would be helping in production of vitamins, right? Like, some of the vitamins, like say that you have vitamin K, your body cannot produce it itself. With the help of the bacteria, these kind of vitamins are being generated in our body. So there are some bacteria in our gut that is good. Okay. So particularly in the gut, in the skin, in the mouth, in the reproductive organs, the microorganisms are found to be living. Okay. Further, we have discussed about the uses of microorganisms also. We have got bacteria like lactobacillus that can be used for preparation of gut, and that happens in the process of fermentation. Fermentation is a process where complex chemicals, biochemicals are converted into simpler chemicals, simpler molecules, right? Simpler substances, basically. Talking about yeast also, we have discussed, it is used in the baking industry to prepare the bread, pastries, and cakes. It basically helps in fermentation. In the process of fermentation, what it will be doing, it will be converting the glucose into carbon dioxide, right? Apart from carbon dioxide, it will be producing the alcohol also. Getting it? So that is how it will be basically useful in the baking industry, and also in the alcohol industry also it is used, right? So it is used for the commercial production of alcohol and wine also. So there basically we will have the natural sugars. Like, for example, we will take the sweet fruits, like sweet fruits would be taken, or we can also take grains like rice, wheat, and barley. So we will take these things, and on these things the yeast will be allowed to grow. So when the yeast will be growing on those sugar rich substances, so it will convert the sugar present in them into alcohol. So that is how yeast is useful to us. Okay. For the very first time, it was Louis Pasteur who discovered the method of fermentation. Okay. Hope you guys are getting it. So hey, you can see that sugar, basically the natural sugars, okay, present in fruits, wheat, barley, etc., will be converted into carbon dioxide and alcohol. Antibiotics we have already discussed, right? Basically the medicines that will help in killing or stopping the growth of disease causing microorganisms. Like you have penicillin. Okay. Penicillin is made from a fungi. Can you guys name that fungi? Yeah. Name the fungi from which penicillin is made. Yes, Chakrika. Yes, Aleph. It's the penicillium. Okay. And who discovered it for the first time? It was Alexander Fleming. Okay. In the year 1929. Okay, we have some more examples like streptomycin, erythromycin, and tetracycline are some other examples of antibiotics. Okay. Then it can not only be used on the humans, but can be used for the plants also. Further, there are some precautions that one must take care of while taking the antibiotics, right? So it should only be taken on the advice of doctor, and one must complete the prescribed course. Okay. If it is taken in wrong doses, so it can lead, it might not be effective, or if the course is not completed, it might not be effective. Okay. And they cannot cure a viral disease. Like if someone is suffering from someone suffering from cold and flu, so taking antibiotics is not going to be effective. Getting it, everyone? So because antibiotics cannot cure a viral disease, a disease caused by virus simply. Vaccines, we have also learned about this. Edward Jenner discovered the first vaccine. It was for smallpox in the year 1798. Okay. So diseases like cholera. Okay. We have one minute, not cholera. Diseases like smallpox. Okay. These can be cured by the vaccine. We have many more other diseases like SARS-CoV-2, right? COVID basically, that can also be cured by the vaccine. Furthermore, we have also learned about different types of disease. So you have got communicable disease and you have non-communicable disease. So communicable disease are basically caused by the pathogens, disease causing microorganisms. So it will be transmitted from one person to another, right? Like it could be malaria, HIV, chickenpox, etc., or it could be typhoid. But the non-communicable disease are inherent, kind of inherent in nature. Basically, they are cannot be transmitted from one person to another. Getting it? So, for example, cancer is a non-communicable disease. Yeah. Cancer is there, leprosy is there, etc. So these are basically non-communicable disease, in short, written as NCD, right? So these diseases could be either due to some genetic factors, right? Could be due to some genetic factors, okay, or could be either due to some allergic factor, or is would be related to the lifestyle or the eating habit of the person. So whereby they are non-communicable, will not be transmitted from one person to another. Also, you can see how the communicable diseases transmits from one place to another. So as the infected, basically as the flies would be sitting on the human fecal matter or animal fecal matter, so pathogens from the fecal matter will be carried onto the body of the fly, and when it will be sitting onto the food, thereby the pathogens from the body will be transferred to the food. When the same food will be eaten by the person is going to become sick, as he or she has eaten contaminated food. Similarly, for the malaria also, we have discussed how it basically spreads. So the infected mosquito would be biting the uninfected person. Basically, the mosquito is said to be infected as the blood it has sucked is infected. It has plasmodium in it. By the way, you guys tell me that the plasmodium present in the blood that is sucked by the mosquito, is it going to affect the mosquito? The infected blood that the mosquito has sucked from the person, will it be affecting the mosquito also?
Yes.
What about others?
So could you repeat?
My question was, no, suppose that this mosquito has sucked blood from an infected person who is already suffering from malaria. So if the mosquito sucks the blood of a person suffering from malaria, will mosquito also be affected by it?
No, sir.
Hmm? Yeah.
I
In
don't
fact,
think so.
it will not. Getting it? No? So the mosquito itself will not be affected by it. It will be basically transmitting it later on to the healthy person when it will be biting them. Okay, good. But I would say, like in certain studies, it is shown that it can change, it can affect them in a way like it can change their behavior. Okay, no, like it can make them more likely to bite the humans. So in that manner, it will affect, but it is not going to make the mosquito sick, but can affect the behavior. Getting it now, Roni, Chakrika, everyone? Okay. Then we have also learned about some particular disease like typhoid, which is cows due to consuming contaminated water. So contaminated water, we have this bacteria called as Salmonella typhi in it. So once the person would be consuming this, so there can be rashes on the body, there can be ulcers in the intestine, right? Then there could be white coating on the tongue, and person will be suffering from the fever also. So these are the symptoms of typhoid, and in order to basically avoid getting infected from this, one must consume clean and pure water basically, right? And the diseases that are spread by the viruses, basically, so far the examples that we have seen, these were the examples of disease caused by the bacteria. Like you have this cholera, you have the typhoid. Okay. And as for this one over here, malaria is a disease caused by the protozoa, that is plasmodium. Furthermore, there are viral diseases also, right? So for example, you have influenza, you have we get to see, like we have influenza, chickenpox, mumps, measles, herpes, etc. So viral disease are infections caused by the viruses, right? And different type of viruses would be responsible for spreading different types of diseases. Okay. One of the most common viral disease is the common cold. Okay, it is caused by I'm sorry. This common cold is the most common type of viral infection that is caused by infections in the respiratory tract. Apart from that, you have got chickenpox, herpes, etc. Then common cold particularly is caused by this virus named as rhinovirus. Okay, some of the names are particularly very important here. Influenza caused by the influenza virus. Okay, then you have smallpox caused by the variola virus. So if you were to mention, remember some of the disease and its causing organism, that would be very good. Fungal disease, we have learned about the ringworm and the athlete's foot. Okay, so we have discussed about that also. Then diseases by the microbes are not only limited to animals or the humans, but it is also spread in the plants, right? So can you guys tell me about names of some of the diseases caused by microorganisms in plants? Try to recall this.
Rust of
Wheat rust or rust of wheat, good. And Mhm.
Puffball.
Repeat again.
Sir, I'm not sure if it's name, but I think it's puffball or something.
Puffball is an example of a fungi now. Puffball, the image which we saw earlier in the previous class, that was a fungi basically. See, anyone remember? Like you might have seen that many
Roll.
times. Mhm.
Leaf roll, something is there. Roll like
Mm, leaf spot.
uh,
Oh, mhm.
leaf roll, something.
Leaf rose. Yellow vein mosaic is there. We have yellow vein mosaic, rust of wheat, citrus canker. These are some of the common examples which we had. So citrus canker would be in particularly infecting the citrus fruits. Citrus canker refer to the citrus fruits. Okay, no? So it is commonly found in the leaves of lemon plant. I had this lemon plant in my home. Last year, it was also infected with the citrus canker. So lemon, as you can see, the leaf and the lemon would be turning like this. So this is the citrus canker, which is caused by a bacteria. Okay. Then rust of wheat is there, which is caused by fungi. Yellow vein mosaic, particularly, was it is seen in the case of ladyfinger, right? So yellow vein mosaic, it is caused by the viruses. Okay. Wheat rust or leaf rust or also brown rust, basically it is caused by the fungus. The name of the fungus is Posinia triticina. Okay. Then as for this yellow vein mosaic, that is basically in this okra. Okay. Basically what will happen, okra would be turning yellowish in color, right? The veins present on the leaf will start to disappear, okay, or it will become yellow in color. Getting it? The stems, sorry, not the stem, the leaf would be kind of getting folded and the growth would be limited. The growth will be basically stunted, like it will stop to grow beyond a certain height. So that is the yellow vein mosaic virus caused by a fungi. Further, we did also discuss about food preservation methods in the chapter, right? So basically, a food is spoiled when there is uncontrolled growth of microbes in it. That is how the food gets spoiled now. So, what is the reason behind sun drying the wheat? Hmm? Or why do we keep the food in refrigerators? Yeah. The reason is basically to create extreme condition or to get rid of the moisture or to create a condition in which the microbes cannot survive, right? So that is all about the food preservation. So we have got preservation by use of oil and vinegar. You can store the food in airtight containers, give it a cold treatment by keeping it in the refrigerator, or basically heat it or kind of dry it in the sun. Certain certain food items like wheat or rice is first dried in the sun in order to lower the moisture content in it. Then fishes can be preserved by the use of salt, and we can also preserve the food with the use of sugar. Okay. So the methods that we have learned can be categorized into two. One, you have the chemical method of food preservation, or one is basically the non-chemical method, where we'll be basically using the traditional methods or would be basically doing some physical changes, like either increasing the temperature, lowering the temperature, etc. So in the chemical method of food preservation, we'll be using some synthetic materials, like chemicals like benzoates will be used to preserve the food. Okay, that is the chemical method. Apart from that, we can also use lactic acid or sorbic acid. That can also be used. Then you have got sulfur dioxide or carbon dioxide. So these chemicals can also be used to preserve the food. Other than that, we have other methods like pasteurization, in which basically the food first will be heated. Okay, then it will be immediately cooled down, right? So this is done to get rid of the microbes present in the milk particularly. Okay, so first they will be heated to somewhere around 70 degrees Celsius for a few minutes, 70 or 71 degrees Celsius particularly for a few seconds. Then it will be immediately cooled down to about 4 degrees Celsius. So first, when you are heating it to this mild temperature, that is less than 100 degrees Celsius, so the microbes will be killed. Further, whatever microbes will be left, that will be cooled down, that will be killed when we'll be cooling it down. So that is how fruit juices and the milk would be preserved. And this method is pasteurization. Who is the person here? Yeah. Guys, tell me who invented the method of pasteurization? Yeah.
Mhm.
It's the same
Yes,
person. Continue.
I think so.
Mm-hmm.
I think so. I will
Oh,
not say.
what about others? I will give you a hint. He's the same person who invented the method of fermentation.
Louis Pasteur.
Louis Pasteur. Okay, now. So that was Louis Pasteur who invented this method. Okay. Then we have methods like salting. You have methods like addition of sugar. Basically, in this method, what happens? The moisture from the food is absorbed basically. So it will create an unfavorable condition for the microbes, hence the microbes will not grow. So traditionally, for a very long period of time, in the olden days, people would be consuming raw meat. Okay, they would be basically taking some salt, would be taking some spices, okay, and will be mixing it in the meat. So meat will be preserved for many months in this manner. Okay, and they would be consuming it in case of famine. Okay, in situations like famine, they'll be mainly consuming the raw and dried meat. Then we have the canning method. Okay, so basically in the canning method, what we see that the food will be stored in tight vacuum packed cans, basically in which air would be removed, particularly oxygen would be removed from that. As we know, oxygen is a basic need for microorganisms also to grow. So we'll be taking out the oxygen and will create an unfavorable environment in which microbes will not grow. So that is the case. As for dehydration, basically, it's the method in which we either completely take out the water from the food or limit the amount of water in the food in order to decrease or in order to stop the growth of microbes like bacteria. Okay, so that is the method of dehydration. In this, basically, the food items can be dried in the sun also, right? So that is the case. As for the freezing, in this the temperature of the food will be brought very low, okay, like between 4 degree to 5 degrees Celsius or even lower than that. So by lowering the temperature, what will happen? The microbes will not be able to reproduce. Their metabolic activity will slow down, and hence they will not be able to grow any further or will might die also. Thereby, certain food items like vegetables, berries, carrots, peas, etc., can be preserved by the method of freezing. Okay, so that was the different types of methods, and the importance of the food preservation is that it can increase the shelf life of the food, right? We can store food for many days. It will prevent the growth of microorganisms, will basically stop the spoilage of the food items, right? And also it ensures that the quality and the amount of nutrients in the food is ensured, that it is not lost. So that was all about this chapter. Any questions? By the way, we have one more topic was there. This nitrogen fixation was there. Yeah, 1 minute. And you guys told me that you had some doubt in this nitrogen cycle particularly, right? Okay, let us discuss this then. Now, as we know that plants requires different type of nutrients. You guys tell me how many nutrients are required by the plants? Is it 17? Is it 27? Is it 30? How many essential nutrients are required by the plant to grow? It's about 17. Okay, na 16 or 17, particularly not 27, Abdullah. It's 17. Yeah. It could be, it is like nitrogen is there, hydrogen, phosphorus, sulfur, magnesium, potassium, calcium, etc. Okay. While some of them are required in very minor quantity, like manganese, nickel, cobalt, chlorine, copper, etc. Now, plants cannot take all of them directly from the water and the soil. To take some of the nutrients, for example, nitrogen, plants have to rely on certain bacteria. Okay, na? So there are some bacteria like you have rhizobium bacteria that will help the plant get the nitrogen from the atmosphere, and they will be doing so in the process of nitrogen cycle. Okay, so in nitrogen cycle, what basically happens, we see that the nitrogen cycle, what will happen, the nitrogen from the atmosphere will be fixed into the soil, and it will go through multiple changes, multiple processes, and will be eventually returned back to the air. It was taken from the air, will go into the soil, will go through some changes, and will eventually be returned back to the air. Okay, so let us understand about this in detail, that what is the nitrogen cycle and what are its steps. So nitrogen cycle is the natural process through which nitrogen moves between the atmosphere, soil, water, and the living organisms. It involves several steps that changes nitrogen into forms that plants and animals can use, and then return it back into the atmosphere. So although we see that plants can directly take oxygen from the air, can directly take carbon dioxide from the air, can directly take the water from the soil, right? But plants cannot directly take the nitrogen from the atmosphere. Getting it, Gauri and everyone?
Yes.
So thereby it relies on some bacteria. Okay, that are basically nitrogen fixation, nitrogen fixing bacteria. So the very first step is the 1 minute. The step of nitrogen fixation. Okay, now the very first step here is nitrogen fixation. That is step number 1. So in nitrogen fixation, the bacteria should be converting nitrogen into usable form for the plant. The bacteria will be converting the nitrogen into usable form for the plant. So bacteria convert nitrogen into usable form for plants. Okay, that is the first step. That is called as nitrogen fixation. Next is the nitrification. In nitrification, what happens? Now the bacteria would be converting this ammonia right over here. This over here is the ammonia. Okay, now. So now the bacteria would be now converting this ammonia into compounds like nitrates and nitrites. Into compounds like nitrates and nitrites. So there are certain bacteria, like you have nitrifying bacteria, that will be converting ammonia into nitrates and nitrites. Is this point about nitrification clear to everyone? What is happening here? This is ammonia, and it is being converted into compounds like nitrates and nitrites. Yes, someone had a doubt. Tell me. Yeah. Someone was saying anything. Okay. So we're saying that this right over here is nitrite, and this right here is nitrate. So in nitrification, we are seeing that nitrifying bacteria converts ammonia into nitrates and nitrites. That is the process of nitrification. Okay. Furthermore, what will happen, as you can see in the diagram, that the nitrates are now being absorbed by the plant. Okay, now, so this is basically assimilation. I think the term assimilation might be familiar to you guys. The chapter nutrition in animals in the 7th grade, you must have learned about different steps of nutrition, right? Assimilation was one of it, where basically the digested food is basically absorbed. So here, basically the absorption of nitrate is basically being taking place, is happening, right? So plants are basically now taking up the nitrogen. Okay. So that is the process of assimilation. So plants absorbs the nitrogen compound like nitrate. So this compound nitrate is now being absorbed by the plant. Okay. Furthermore, some of the nitrate, what happens to some of the nitrate is now being converted back into the gaseous state by the denitrifying bacteria. So here you have the process of denitrification, where this nitrate, that is NO3, will now be converted into N2, that is the gaseous state of nitrogen. Tell me if this thing is clear so far to everyone.
Yes, sir. Clear.
Clear? Good. So in denitrification, basically, in this one particularly, we'll come back to this ammonification in 1 minute. Denitrification, the nitrates are converted back to atmospheric nitrogen, meaning that the nitrogen in the gaseous state back to atmospheric state. There was one more step here, that is ammonification or basically decomposition. So what is basically happening now? Like when the plants and the animals would be dying, so when their body will decompose, so in the process of decomposition, the nitrogenous compounds that were locked in their body will be released by the action of decomposers. Getting it? So that is the process of ammonification basically here, where when they will be dying, 1 minute, when they will die, so the nitrogenous compounds locked in their body are released back into the soil, and this is the compound called as ammonia. Okay, so that is ammonia or decomposition. So when dead organisms decay, then nitrogen in their body is released as ammonia. So that was all about this nitrogen cycle process here. So as you can see here, the nitrogen from the atmosphere was taken, and now via different steps, it is released back into the air. And this nitrogen cycle is very crucial, as nitrogen is one of the 17 essential nutrients required by the plant. So hope that much should be clear. Okay, so that's it for today's class. In the next class, again, we'll be taking up some questions from subjective questions from the chapter Age of Adolescence. Okay. Okay, then bye everyone. Gauri will be staying. By the way, only Gauri you are having this chapter, Chandrik 5%